Hello, everyone around the world. I'm Arsalan, and today I want to talk about how the bacteria in our body can affect cancer and can fight cancer in our body. And the article I'm talking about is based on the research in Chicago Medical Center, and it focused on this question: what if the bacteria living in our gut could actually be engineered to fight cancer? And that's a good question. Maybe we don't know how the bacteria in our body is important to our body, and what are their rules. It may sound like science fiction, but scientists now working on exactly this idea. Researchers at the researchers at the University of Chicago have engineered a probiotic bacterium that can enter pancreatic tumors and act like tiny drug factory. The bacterium is designed to produce an immune stimulating molecule directly inside the tumor. And in animal studies, this approach showed the growth of pancreatic cancer. Even more interestingly, the treatment appeared to work better when it was combined with chemotherapy, radiation therapy, and immune therapy. So, how does this actually work? Let's take a closer to look. Why pancreatic cancer is so difficult to treat? Pancreatic cancer remains one of the most challenging cancers to treat, and one major reason is not simply the cancer cells themselves, it's the environment surrounding the tumor. You can think of a tumor as more than just a mass of cancer cells. Around those cancer cells, there is a complex environment made up of blood cells, connective tissue, immune cells, signaling molecules, and many other components. Scientists call this tumor environment. And in pancreatic cancer, this environment can be um, particularly um, hostile to the immune system. In many cases, the tumor creates what researchers describe as cold tumor microenvironment. In simple terms, immune cells has a much harder time entering the tumor and mounting an effective attack. And this is one reason why some forms of immune therapy that work remarkably well in other cancers have had much more limited success against pancreatic cancer. So, researchers at the University of Chicago wanted to find a completely different way to bring the immune system into the fight. The surprising idea is use bacteria as a drug delivery system. The researchers turned a bacterium called um, Bifidobacterium longum. You might have heard this before. This is a type of probiotic bacterium that naturally occurs in human gut. But in this study, scientists didn't simply give patients a probiotic, they genetically engineered the bacterium and then gave it a very specific job. The engineered strain is called Bifido SUMIL2. Its mission is basically this: to find the tumor, enter the tumor, and then produce an immune stimulating treatment right there. So instead of giving a drug and hoping that enough of it reaches the tumor, researchers are trying to use the bacterium itself as a delivery system. That's why the researchers describe these bacteria as tiny drug factories. But why would the bacteria go to the tumor? This is probably one of the most interesting parts of a research. This bacteria has a biological characteristics that makes it particularly interesting for this purpose. It's an obligate anaerobe. That means it prefers environments with very little oxygen. And many solid tumors contains regions where oxygen levels are extremely low. This includes pancreatic tumors. Healthy tissue, on the other hand, generally have more oxygen. So, when these engineered bacteria are introduced into the body, the researchers found that they can uh, preferentially accumulate in the low oxygen regions of tumors. In other words, the tumor can provide an environment where these bacteria are able to become active. And once they are inside the tumor, they can start producing therapeutic molecule that gives scientists some very interesting a living delivery system that can potentially concentrate a powerful treatment where it's needed most. So what exactly is the bacteria producing? This is where immunology comes in. The engineered bacteria are designed to produce a modified form of a molecule called interleukin 2. Interleukin 2 is an important signaling molecule in the immune system. One of its major roles is to stimulate certain immune cells, including T cells. And T cells are among the key players responsible for recognizing and attacking abnormal cells, including cancer cells. But there is a problem. Giving interleukin 2 systematically can cause serious side effects. And conventional interleukin 2 can also stimulate immune cells called regulatory T cells or Tregs. These cells can suppress immune responses. So, researchers wanted to make interleukin 2 more precise. They developed a modified version called SUMIL2. The goal was to stimulate cancer fighting T cells more effectively while reducing stimulation of regulatory T cells. Then they put these modified molecules inside the engineered bacteria. So now you have a bacterium that can potentially enter the tumor and produce the immune stimulating molecule locally. Instead of releasing large amount of treatment throughout the entire body, the idea is to concentrate it inside the tumor. What happened when they tested it? This is where the study becomes especially interesting. The researchers tested this bacteria IL-2 or Bifidosumil2 in animal models for pancreatic cancer. They found that the engineered bacteria preferentially accumulated inside tumors. They also observed increased immune activity within the tumor. In particular, the treatment increased the activity of CD8 positive T cells. There is important cancer fighting immune cells. And as the immune response became stronger, the growth of pancreatic tumors slowed down. So the bacteria were simply sitting inside the tumor. They were helping change the immune microenvironment around the cancer. And that could be particularly important for pancreatic cancer, where the immune system often struggles to gain access to the tumor effectively. But the researchers didn't stop there. They asked another important question. What happens if we combine this bacteria therapy with treatment that were already being used against cancer? So they used combination with chemotherapy. They also tested it with radiation therapy, and they tested it with immune therapy, including anti-PD-L1 treatment. And the results were even more interesting. In the animal models, combination of this bacteria SUMIL2 with this established treatments produced better tumor control and longer survival than the individual treatments alone. This is important because the goal may not necessarily be to replace chemotherapy or immunotherapy. Instead, this technology could potentially make existing treatments work better. Think about it this way: chemotherapy attacks cancer cells, radiation damages cancer cells, and immunotherapy helps the immune system attack cancer. And the engineered bacteria could potentially help create a more favorable immune environment inside the tumor itself. So the bacteria could become another tool that works together with existing cancer treatments. So this makes a cold tumor hot. This is one of the most exciting concepts behind the research. Remember the term cold tumor. It basically describes a tumor where the immune system isn't mounting a stronger enough responses. So it means researchers are interested in finding ways to turn these tumors into what are sometimes called hot tumors. A hot tumor is one where immune cells are more active and more capable of attacking cancer cells. The engineered bacteria could potentially help with this process. They enter the tumor, they produce SUMIL2, and this bacteria stimulates cancer fighting T cells. And these T cells can then become more active against the cancer. So the bacteria aren't necessarily killing the cancer directly. They are helping recruit and activate the immune system inside the tumor. That's very difficult way to thinking about cancer treatment, but there is a very important limitation. Now, this is where we need to be careful because this is not yet treatment for people. This research have so far tested these bacteria in animal models. It has not yet been tested in humans, and that's huge distinction. A treatment can look extremely promising in mice and still fail during human clinical trials. Human biology is much more complicated. So, before this approach will become a real cancer treatment, researchers need to answer several important questions. Is it safe for human? Can the bacteria stay where they are supposed to be? Could they affect healthy tissues? How long does the immune responses last? Could the bacteria cause unwanted inflammation or infection? And can researchers control bacteria precisely enough? These questions still need to be answered. The team also wants to investigate where the bacteria could eventually be delivered orally instead of through injection. And there is another misconception we need to avoid. When people hear the word probiotic, they might immediately think, can I just take a probiotic supplement and prevent or treat pancreatic cancer? Absolutely not. That's not what this study showed. The bacterium used in this research was genetically engineered. It was specifically designed to produce a therapeutic molecule called SUMIL2. So, eating yogurt or taking a conventional probiotic supplement is not equivalent to receiving this experimental therapy. So, exciting part isn't simply that this bacteria exists in gut. The exciting part is that scientists are learning how to engineer bacteria to perform a specific therapeutic function. Um, and this is the way that more researches should be done in this way, and it needs more study, more human studies to assess exactly um, can engineered bacteria help the treatment of cancers, different types of cancers. This study was for University of Chicago Medical Center and reported by Science Daily in 2026 August. And the original research was published in Science Advances under the title Engineered Probiotic um, Bacteria for Tumor Targeted Pancreatic Cancer Therapy. And the bacteria is Bifidobacterium. And if I want to spell it, Bifido, and then bacterium. And please, if you liked this video, like, subscribe, and because I read hot topics in medicine and yeah, also cancer area, and it's very good for me if I find viewers around the world that are enjoying my contents. Bye.